a plus side post-test. We get to kind of start fresh. That's always sort of nice. We will be starting the life in the water, which is really looking at, as we think about the first sort of set of organisms, and we've already started to look at these in lab. What are some of the basic traits, right? Basic phenotypes, basic behaviors that you need in order to be successful as an aquatic organism. And so we're going to look at some very general ones, some of which you've seen already and some of which we'll see as we look in more detail at our organism classes. We're going to kind of walk through these in large groups, looking at things like movement, respiration, reproduction, and think about, well, what are these things that allowed these many classes, right, from our early fishes to chondrichthyes to osteichthyes, to be so successful as aquatic groups as a whole, right? What kinds of things can we be looking for? Okay, so as I mentioned, we have some sort of like larger categories within here that we're going to be working in, right? Respiration, or the ability to breathe, which we know is going to be a little bit different. Buoyancy, which we've talked about on and off in lab, right? How do we float or adjust our height within that water column? Okay, sensory. We know this is going to be a little different. So how do we experience sensation? Right? As land-dwelling animals, we usually think of this as like hearing and touch, and we can certainly imagine when you're embedded in water, like when we're underwater, that changes drastically for us. Right? Our hearing sucks underwater. So we know if fishes are going to be successful, that they need to be doing these things certainly a lot differently. Okay? And lastly, we're going to talk about Osmo Regulation, which just literally means saltwater balance. Right? This is something that's going to come up again when we talk about land. We want to think about if you are, for example, a fish completely submerged in a very salty environment, how do you maintain whatever it is you need, right, your own water, without becoming super salty yourself? Okay, so we're going to start moving on through, and of course, like all my roadmaps, we're going to start at the beginning. So let's start talking about respiration. So as we might imagine, respiration underwater is going to be rife with its own set of challenges. Okay, specifically, right, because we're thinking about breathing water or using water as our respiratory substance instead of what we kind of conceptually like to biasly think about, which is air, right, what we use. Now, water is its own problem, largely because of these three pieces. First of which is water is not naturally as oxygen rich, right? The air we breathe is 20% oxygen. It's mostly nitrogen, but at least there's 20% oxygen in it for us to use. Water is significantly less than that, right? It's an order of magnitude lower by content, normally, on average. So there's already less for them to be working with. Right? Intuitively, water is very viscous. Right? Viscous is talking about kind of how thick the material is, right? So air is very thin, moves around freely, okay? Both in when we move through it and when we're trying to move it through our body, right? It's very easy to move around because there's not much substance to it. Water, of course, is going to be the opposite problem. It's very viscous, relatively speaking, to air, right? It's much thicker. And so it's much more energy costly to move. It's harder to move around in, right? Remember, we talked already with body movement about how if you're going to move around in water, we're going to deal with that by being more fusiform. So there's going to be additional challenges for sucking that in, right? It's going to be harder to move that into our mouths and into our respiratory organs. Okay, and then when you put these two combos together, the fact that water is very thick and viscous and naturally oxygen poor, that dispersal of oxygen in water is not going to be even, right? So if you imagine, for example, like a pond, okay, the top of that pond is going to have a higher oxygen content, right, where it's interfacing with that air, so you can get nice exchange. There'll be diffusion happening where the oxygen and the air is coming down to those top layers of water. But those much deeper layers of water, okay, are not going to mix as easily, right? They're going to be very deep. They're not actually touching any air themselves. So they're going to have to sort of assume that the air, that oxygen, is going to trickle down from the very top of those layers, which it will to some degree, right? But it works like any other diffusion, right? If I spray perfume up here, right, my first row is probably going to smell it really well. You guys don't smell anything, but right, normally you'd be able to smell it pretty heavy. But my back row, okay, being the deepest part of the water, okay, maybe you'd be able to smell it, right? Because by the time it gets back there, right, it's already had to cross so much space, there's not going to be much of it left, okay? Now, if we consider something like a river, right, that's splashing and mixing and churning, literally swirling in water, something like that might have some more oxygen. So it's going to be very water dependent, okay? But in general, it's very hard, right? It's very hard to get oxygen into water, and it's super hard to get oxygen into water evenly. So fishes have a very specific challenge, or anything aquatic, right, has a very specific challenge on getting a good amount of oxygen efficiently. So they need to make sure whatever system or systems, as we'll find that they use, need to be very efficient because they have to use what they get. Okay, does this make sense so far? Okay, so these are the problems that we need to overcome. Okay, so there are a couple of other issues that we're going to cope with. The deal bit of gas solubility. In other words, how easy does the oxygen actually mix in that water? Okay, so we already talked about this first bullet, right, which is basically just the law of the diffusion. Right, we're going to get more oxygen, right, the closer it is to anywhere where there is air. Okay, so this is like that top of the pond business. Okay, you'll be able to pull more oxygen in when you're exposed to more oxygen. Okay, this is my front row, getting smacked in the face with that spray of perfume. But there's two other things that may increase the ability, or decrease is appropriate, right, to get oxygen. Okay, this first one talks about temperature. Okay, colder water has higher temperature. Colder water has higher oxygen overall. Okay, and so as we slowly increase the water, and we decrease the water's ability to hold oxygen molecules. Okay, this is one of the reasons that you see like small warm ponds get what we call stagnant. You pretty much only see flies and algae sit around on them. They kind of smell a little, right? That's because they're dead water, right? There's not a lot of oxygen in them anymore, so there's not a lot of animals that can live in that anymore. Okay, the other thing that happens is the saltier the water. Okay, now salt can mean a lot of things. We can think about it as regular salt, but this can be anything, any solute we put in the water. All right, so you see I have several other examples on here. For example, a lot of fertilizers in the water. Okay, for example, any of those kind of compounds, nitrogens, phosphoruses, right? Anything that's going to add to that solution. Can I think about cooking, right? What kinds of things can I dissolve in a water? So, anything that we dissolve in that water, okay, is kind of competing for space in that water. So the more other stuff that we dissolve in that water, okay, the less ability oxygen is also going to be able to dissolve in that water. Okay, so the best kind of water is going to be like a cold freshwater river, right? It's going to be mixing a lot. You're going to be able to hold that oxygen a really long time. Okay, the worst kind of water, right? It's going to be a small, solute filled, maybe it's got a lot of runoff or it's a little salty, right? Warm pond, right? Something that's not mixing, okay, it's quite warm. And so we've got temperature and these salts competing with oxygen, right? So it's going to have a much lower oxygen content. You can kind of see two ends of those spectrums, right? And of course, there's anything in between. I still good? Okay. So let's start to look at how exactly are organisms overcoming these issues, right? And so remember, because we have such large issues, our goal here is going to be very, very efficient at making sure that we can extract oxygen. So the first thing we're going to talk about is the gill structure on fishes, right? This is where oxygen exchange is actually going to occur. Okay, so we're going to get a chance, if you didn't, make sure to look at gills last week in lab on our sharks. You're going to get another chance to look at them this week in lab in our osteichthyes. So one of the things we're going to see as we're talking about organisms is the main theme is always increased surface area. We saw this a lot, what feels like back in the day, right? We talked about things like inside the cell. Remember, like the mitochondria had cristae, increased surface area, right? That makes the inside of your cells work really well. The same concepts apply when we're talking about anatomy. So our goal is still increasing surface area for a lot of our organs. And anything that allows us to increase surface area still makes that thing better at whatever it does. So whatever feature increases that surface area is going to make it most effective. Okay. So remember when we were talking about our upgrades, okay, our vertebrate upgrade was having this gill arch. And this becomes very important because this is going to serve as the structural piece that all of our surface area can then build on. So our gill arch, blue kind of stinks. Let's try something that maybe will stick out. Everything kind of stinks. Is this piece here? So we're going to do this bit by bit. And as the name implies, this is fundamentally just a structural component. Okay, so we're going to have multiples of these, as we can see on our picture here, going down either side, and this would be repeated on the other side, right, of our fish. And this is where each of our gills are going to start, right? So we have a hard structural component on which to build our gills. So from there, where that gas exchange is actually going to occur is on what's called the lamellae, right, which are those words there. So we have primary and secondary lamellae. Okay. Okay. So my primary lamellae are going to come directly off the gill arch. And that's kind of what you can see here. Right? They look a bit like these kind of pink fingers. So go ahead and trace these out. I'm going to do a few so my drawing doesn't get too huge. Okay, so these are, and they are red or pink on the fish, right? So these are blood or capillary filled vessels, okay, through which our gas exchange will start to occur. Okay, but remember our key is surface area. Okay, so having a whole bunch of primary lamellae is good. Okay, but we can do better. And so this is where the second lamellae comes in, secondary. What color should we use? See if I can get this like hot pink to show up. So the way we get a lot of surface area and where our gas exchange actually occurs is in the secondary lamellae. So if we imagine our primary lamellae being like long fingers coming straight out of our gill arch, our secondary lamellae then are like hairs on the fingers. And they would be everywhere all alongside them. Trying not to clutter the drawing too terribly much. Also because my drawing skills are questionable. So on the actual image, right, these would be like the lines that they have. Okay. So we can see now, because we basically have structural arch, little fingers, which are going to increase all of the surfaces. And now all of these little hair type things, okay, along those finger-like structures, there's lots and lots of space on which gas exchange can occur. Oops. Okay. So that's step one as we're thinking about this. Okay? We have an anatomical structure that's so starred out, right, branched out, okay, that we have as much surface area, as much space as possible to exchange the carbon dioxide from my body for whatever oxygen is present around me. And so that's one way I can sort of deal with some lower oxygen content space. Okay, any question about gill structure?
Does gas exchange only occur on the middle?
Correct, yes. So gas exchange is occurring on the secondary lamellae. And despite my poor drawing, right, those secondary lamellae would be quite packed, right? Wouldn't have like half an inch between them or whatever.
Does gas exchange not happen on the primary end? Like it's not as much?
So it's not occurring, right? So all of the capillaries, the primary or the secondary lamellae is loaded with capillaries, and that's where we're seeing this occur. Shiny? Okay, so let's see what's actually happening on our lamellae. So as we think about what's happening, when we're actually exchanging oxygen, okay, we want this to be as efficient as possible as well. And so the key here is to make sure that for every second, every moment, that my capillaries, right, my lamellae, are encountering water, okay, I am able to actually exchange, continuously exchange my carbon dioxide, that's what I want to get rid of, right, and then take in the oxygen that's in the water. Okay. So the way we do this is called countercurrent exchange. And of the two images you see here, right, that's the bottom one. Okay, so we're going to walk through how this works and why the system works the way it does. Okay. But that's the big key here, right, is this last bullet point. A. as we do this, we want to make sure that as we're touching, okay, I'm able to continuously make that exchange. Okay, so let's just focus on the bottom image for a second. And work through how this works out. Okay, so in this image, okay, this bottom one is going to be my body. Okay, so this is the capillary that's got blood in it from my body. You know, so this is obviously somewhat exaggerated. But so we can assume for the sake of argument that I'm starting at like 0% oxygen. Very exaggerated. Certainly we never get down to 0, right? But it would be very low, right? By the time that my blood gets around to my gills, it's traveled through my body, it's donated oxygen to all the places that's important, my brain, my heart, my muscles, making all my fishy body happy and worky. So by the time it gets around to my gills, the whole point is I want oxygen. So, right, it's come to and it's very low. That's terrible handwriting. So that's what we mean by that. And so the flow of my body is going one way, or my blood. Now, the top bar here is the flow of the water. Right, that's where we're getting our oxygen from. So this is my water. Okay, here's the start of my water. Right, because we're flowing this direction. This is one of the keys for flowing opposites. So you see when the water enters by my gills, right, it has high oxygen content. Right? For the sake of argument, it's just opposite, right? It would come in as high as it's ever going to be, whatever that means. Right? So it hasn't given me any oxygen yet. It's just now entered my gills. So we're going to make this simple and easy. So it's at 100% capacity, right? It's not passed on anything yet. So we are full. Does everybody understand that setup? Okay, so this is our key when we say countercurrent. Okay, this is referring to that opposite of flow, right? My body or the blood is flowing one way. The water as it comes in is flowing in the opposite direction. And then that big key here is seeing my water's coming in full, my body is coming in empty, right? Ready to refuel. Okay, any questions so far? All right, so as we go through this, pink is not going to show up very well. What can we use? Green hasn't put me down yet. All along the way, okay, the idea is the water is donating oxygen to my body. Okay. So since my body is at 0, okay, or very, very low, okay, whatever we want to imagine here, okay, the key is our body oxygen is always going to be lower, and this is what we want, right, lower than the oxygen content of the water. Right, because we're going to be flowing, right, we got to remember some old diffusion, right, rules. Right, remember solutes always flow down their concentration gradient. So we want the oxygen, and I'm using green, which I know is a little counterintuitive, but I feel like blue is not going to show up great on my image either. We always want the oxygen to flow down its concentration gradient. All right. In other words, I always want, at every moment here, at least some oxygen to come from water to body. Okay. Now we know at the beginning here that it's gonna, because I'm at like 100% water capacity. Okay, so no matter how full my blood gets of oxygen, right, I can't be higher than whatever that oxygen capacity of the water was to begin with. So I can be close. Okay? But you can't be the same. As the water is flowing, as the water is flowing this way, right? I've given up a little bit of oxygen. Maybe I have less. Okay. The numbers don't matter, right? We're just understanding I've given oxygen at the last step, so I have lower. Okay, but this blood here is going to be lower too. Okay, so I'm still going to be able to give whatever. Okay, onward and so on and so forth. Okay, so even when we're down at the lowest, making numbers up. Okay, so let's just say, okay, we've given up all the way. Okay, when we get to the bottom, okay, our oxygen from our water won't be at zero, right? We won't be completely out. We won't be totally done. So we'll still have the ability here, okay, to give some oxygen, okay, to our starting lineup. So as we look along our gradient, right, the big key here is every step along the way we've been able to give, okay, even though these gifts aren't huge, right, these aren't big exchanges necessarily, right, from 100 to 90, but we've given a little every step of the way, and it's been additive, right? This was at 0, then it was at, I don't know, right, 20, 30, 60, whatever it is, right? So we've been able to build up the oxygen content of that blood little by little as it's flowed by. Okay, by being able to continue to add to that blood every moment that it's touched. It's kind of like a parade, right? You never get a lot of candy when you were a kid when they would like toss, but you get like a piece or two. So by the end of the day, you'd have like a nice little handful, right? Or like trick-or-treating. You don't get a lot from one house, but the key is as you keep going up the street, right, every house you touch, you get a little bit more from. And that's what we want to be happening here. Every time that we touch something, we want that exchange to occur. Okay, until we have as much oxygen as we can possibly hold. Right, because then we have to go through the whole body again to redistribute it. Okay, everybody see how countercurrent works? Any questions about that? Because I'm going to compare it to concurrent, so we see why that doesn't work.
So this happens in the secondary lamellae?
Correct. This happens in every single secondary lamellae. So there's lots of locations that this can happen with, which is very efficient. And then this system itself is very efficient, right? We're trying to jack up the blood as full as we can at every opportunity that we can. Okay, problem with concurrent. Okay, so this is the image we see on the top. Make myself the teeniest bit of space here. So concurrent literally means same. So as opposed to our blood and our water flowing in opposite directions, okay, the system we have up here has been flowing in the same direction. All right, so here's my body, right, starting out at pretty much nothing. And here's the water, okay, starting out pretty much full. Okay, so the start of this looks like it's going to be a super great deal. Okay, right? As I have 100 or very full for my water, and I'm very, very empty for my body. Okay, when they're first meeting, okay, we're still going to be working down our concentration gradients. Okay, all those rules still apply. So we're certainly going to be moving oxygen from the water to our body. But here's where we're going to run into a problem. As this happens, these are very large differences. Okay, 100 to 50, or 100 to nothing, right? That's a big difference. It's a big exchange. Okay, so we're going to see right, just like when we used to do this in diffusion, right, a bigger error, indicating a bigger movement of solutes. Okay, so as we think about the next step of this, okay, I have a lot less. Okay, not so bad. Water is still bigger. In fact, still a lot bigger, so we'd do that again. But as we can see at the bottom here, as I reach the bottom, we're going to run into a problem at some point. Okay, and a problem that's pretty high up. As we approach this 50 mark, that's a 5. Whatever. The math didn't quite work out this time. Make magic numbers. Okay. 1, we're going to be able to pass on less and less until eventually we're about the same. Now we reached about the same in countercurrent exchange at no point. Right? We were always higher in the water. Right? So we're always able to give. More so when we were kind of close down here, right, or up here, like when we left, we were near 100. When the blood is leaving the same up here, we're near 50%. So our blood is not going to be able to get full. Right? Even though our first couple moves, we're able to dump a lot at once. It seems like, well, this worked really fast. Right? Because our body filled up on oxygen, then we become equal on the right-hand side there. And that's bad. Okay, so we don't have as an efficient a fill-up of oxygen for our concurrent flow system. Okay, no, that's it. I thought maybe my watch was off. Does everybody see where our big problem here is? Now, this system is super common for like heating units. Right, so your like register in your house commonly works like this, right? Because I want my house to be a very specific temperature, and it wants to be the temperature of the heat that it's kicking out. And so we would want those to match at not a 100%. So those work. Okay, but it doesn't work when I want to like super load something, which is what we want oxygen to do, so I can carry it around my body to redistribute it other places. Because remember, our goal here is to be ultra efficient. Water is hard to work with. So we want the most efficient way possible to do something, and countercurrent exchange is the most efficient way. Right? We can get the most bang for our buck, right? The most oxygen left in the blood when we're finished. Okay, so this involves sort of wiping off a lot of that diffusion dust. Okay, any questions on how we kind of work through that together? Okay, so when in doubt, think about if I had to set up a concentration gradient at the front or the end, like where the flow's coming in or the flow's going out, would I still have oxygen exchange? Right, that's the big key. And if the answer is yes, then you have a good efficient organism. If the answer is no, right, no exchange is happening here, then we have a problem. Okay, any questions about countercurrent exchange? Okay, this is very generally between the Gill Arches system and countercurrent exchange, how all gills work. So what we want to talk about next is how exactly do you get the water to the gills? Okay, so the gills are going to be the site of actual oxygen exchange, and we have to get the water across the gills in the first place, right? So the fresh water containing the oxygen can actually make that exchange with the very needy blood in the fish's body. And so in order to do this, we need to move that water across the gills. Okay, we do this in a very specific way, right? Anterior to posterior. Oops, sorry about that, everyone. So we're practicing our terms. Okay, so anterior means front, and posterior means back. In other words, we're moving from the mouth to back behind the body across the gills, right? This is a unidirectional movement. Okay, and the key here is we need to consistently be moving fresh water over the gills, right? Because remember we said that the water itself is not going to consistently get fresh oxygen because it's viscous. Oxygen is not consistently moving in the water. So we need consistently to get a different spot of water or new water to make sure that we're getting that oxygen. Okay, so how can we do this? There's two major ways an aquatic organism can do this and be successful that we'll see, and it depends on the group or sometimes the species. Okay, the most common is an active pumping system. And that's what I have up here. Let me erase this really quick. Okay, you've probably seen this. Okay, sort of like what you see when the fish looks like it's swallowing or gasping underwater. Okay, it's literally breathing. So this is sort of a two-step process. So what we see here, step one, right, which is my left image, is the sucking step. Okay, so we literally see its mouth open, it's swallowing a bunch of water. Right, so this is the water going in through the mouth. Okay, in step two, okay, we see that that mouth is closed. Okay, and any spaces, films, or this week you'll see bony plates called a perculum, okay, that cover the gills are opened, okay, allowing this mouthful of water to get pushed out both sides. Okay, so I take a mouthful of water, swallow the water. Mouthful of water, swallow the water. Okay. Now they do have two different swallows, right? Notice we do have a throat here. So we're not swallowing our digestion. Okay, we're literally pushing them out our gills. Okay, but it comes as naturally to them as the breathing that you're doing right now, right? This is not something they're largely thinking about. So most of the fish that you're thinking of do this, right? Pretty much all of our bony fishes, trout, guppies, clownfish, the whole thing, right? Some sharks also do this. Okay, pretty much if you're gonna sit still and hang out, okay, you're gonna have to do this. Okay, pretty much sucking the water, right, keeping it moving forward. You're diffusing water into your body actively. Kind of keeping a small flow going, a small stream. What happens when I just do this? The second option that occurs, and we still need that top thing to be occurring, right? Our goal is still to move water anteriorly to posteriorly and constantly move that water, but not all fish can or do effectively pump. And so they use what's called ram ventilation. Finally, something that's named intuitively, at least. Okay. So this is really common in most sharks, particularly larger sharks. Okay. So remember from last week, right, in particular sharks and other cartilaginous fishes, okay, as well as things like our lamprey and hagfish and all that do not have any sort of casing over their gills. So pumping gets a lot harder because you can't make that seal in step one, right? So what's more common, particularly since some of these guys get quite substantial in size, right? We can picture some, I cannot talk, picture some quite large sharks. So what these guys tend to do is literally ram, right? So as they're swimming, water enters their mouth. They just swim with their mouth open. Water will enter their open mouth. Okay, they keep the back of their throat closed. And so as they swim forward, the water has nowhere to go but out and over the gills. Okay, now the benefit of this is a pretty low-tech hack. As far as breathing goes, I'm just going to swim around with my mouth open and everything else is just going to do the job for me. Okay? The downside is what happens if I stop? I can't breathe, right? So I always have to move. So a lot of these larger sharks, need to keep moving to survive. Any sharks, like a nurse shark or a lemon shark, for example, that is to my password, that are a little bit smaller and spend some time sitting on the ocean floor, can do both ram ventilation and pumping. Okay, larger sharks like the great white that only ram ventilate, they can't stop, not for extended period of time, right? Because they're literally holding their breath anytime they stop. Okay, my back two rows, when you are all completed, you are welcome to go.